Our first speaker of this session is Joshua Jenkins from Leeds. Josh did his PhD at the University of Bristol, working in electron tomography and volume EM, before doing a postdoc in Canada. Since 2023, he's been back in the UK, working as a research fellow in structural neuroscience in Renee Frank's lab in Leeds. And that's what he's going to tell us about today.
Thank you for the introduction. So today I'm going to show the first visuals of amyloid pathology resolved by cryo-ET within the context of Alzheimer's disease, postmortem human brain tissue. Just going to start with a quick primer to neurodegenerative disease and amyloid. So most neurodegenerative diseases are associated with the deposition of a disease-specific amyloid protein. And over time, these polymerize to form fibrils and filaments inside the human brain. On this slide, you can see the structure of several amyloid proteins, which have been determined by extraction from human brain tissue and using helical reconstruction methods. If we look at Alzheimer's disease, we can see that this is characterized by the deposition of extracellular beta-amyloid plaques, which comprise of A-beta fibrils. It's also characterized by the deposition of intracellular tau, which forms inside neuronal cell bodies, and dendrites, and these contain straight filaments and paired helical filaments. While we have the high-resolution structures of some amyloid proteins, we wanted to determine what the structure and organization was directly in human brain tissue, so we're retaining that cellular and tissue context. This slide shows a high-level overview of the cryo-clem workflow that we developed and utilized in order to do this. So we start by receiving frozen post-mortem human brain tissue from the Netherlands Brain Bank. And this is frozen by rapid autopsy. So within a few hours, a piece of that brain tissue is frozen. It's then shipped to us. We thaw it to room temperature, and we prepare acute slices of the brain tissue using a vibratome. These are approximately 150 microns in thickness. And then we incubate the brain slices in a fluorescent amyloid dye, methoxyX04, to label the regions of amyloid pathology. We then vitrify using high-pressure freezing, and we can use cryofluorescence microscopy to actually identify within those acute brain slices where the amyloid pathology is. And as we've heard in previous talks, we can then use techniques like cryofibsem and Semavis, or cryo-ultramicrotomy, to prepare even finer cross-sections through the tissue, which is suitably thin for cryo-electron tomography. So if we start with cryo-clem-targeted cryo-ultramicrotomy, this approach enables us to generate brain tissue cryosections that are approximately 100 to 200 nanometers in thickness, targeted at regions of amyloid pathology. So we can see on the left-hand side, these are cryofluorescence images of entire high-pressure frozen carriers containing the vitreous brain tissue, which have been incubated with methoxyX04. And in the middle here, I'm just zooming in on these regions to show the beta-amyloid plaque pathology, the tau tangles and tau threads, which we can then prepare cryosections from and transfer to cryo-EM grids. And you can just about make out the repeating fluorescence here. So what can we do with these approaches? So on this slide, we can see the first glimpses of a beta-amyloid plaque resolved by cryo-ET within the context of vitreous Alzheimer's disease post-mortem human brain tissue. So this is a tomographic cross-section taken through a region of a beta-amyloid plaque. On the right-hand side, we can see these high-contrast dots. These are A-beta fibrils, which are running axially through the tissue. We can also see faint filaments running in the top left and in the middle. And then interspersed amongst this, we've got non-amyloid constituents. We've got extracellular cuboidal particles, membrane-bound compartments, extracellular vesicles, and droplets. And I think what's really interesting about this is if we look at the segmentation on the right-hand side, the A-beta fibrils are highly branched. So it suggests that there's a growth mechanism occurring by secondary nucleation. We're now looking at a tomographic cross-section taken through a myelinated axon. So you can see the multiple layers of membrane. And in the middle here, we've got, again, high contrast dots, but the diameter, the cross-sectional diameter is slightly larger. And these are parallel clusters of unbranched filaments. So this is an example of an intracellular tau thread. We also found examples of extracellular tau, which likely have remained following neuronal cell death, because they're not contained within a membrane. Again, these are parallel clusters of unbranched filaments. Here we can see this cluster weaving around membrane-bound compartments and situated between a myelinated axon and a mitochondria. So once we'd looked at the in-tissue molecular architecture of these different pathologies, we wanted to try and confirm the identity of the proteins directly in human brain tissue. So we developed a subtomogram averaging pipeline based on the WARP-M RELION. So in the top left, this is the tomographic cross-section that I showed on the previous slide of an extracellular tau. And in the middle, I've just highlighted cross-sections through 5 filaments. If we rotate the tomogram 90 degrees, we can see the helical axis of each of these filaments. And we can model these and provide them as initial coordinates for alignment and averaging of whole filaments to get a better estimate of the crossover distance of filaments, and from which we can determine helical twist. To then try and obtain higher resolution structures, we can position additional model points along the helical axis, and then using that kind of repeating nature of an amyloid, we're increasing the number of subvolumes that we can use to generate an initial model. And we can feed this into RELION and perform helical reconstruction. And using this approach, we were able to resolve the polypeptide backbone conformation and inter-beta sheet separation of paired helical filament tau to 8.7 angstrom resolutions from 1 tomogram in tissue. And to try and objectively validate the quality of this map, we used Phoenix EM Place Local, developed by Randy Reed and co-authors, to quantitatively score the fit of the paired helical filament atomic model into the subtomogram averaged half maps. And it's generated an LLG score of over 1,000, which is highly significant. So at these sub-nanometer resolutions and also at more intermediate resolutions around 20 angstroms, we can begin to accurately determine the polarity orientation of tau in tissue. And we are interested in this because we know that polarity orientation is functionally important for some filamentous proteins. A good example are microtubules. So in neuronal axons, microtubules have a highly uniform polarity orientation, whereas in dendrites, it's more mixed. For tau, it could be biologically significant and possibly tell us about biogenesis of these filaments. If we imagine when a tau filament is polymerizing, at a certain point, the length of the tau filament will exceed the dimensions of the width of the compartment within which it's growing. So its ability to flip polarity will be restricted. So it must become fixed at an initial stage of assembly. And by quantifying it, we can possibly glean information about how these filaments are nucleating and then spreading. So we set out to quantify polarity orientation, but also to look at the probability of the splits that we would get in those clusters. So if we look at the extracellular tau, which achieves sub-nanometer resolution, this contained 136 filaments, this cluster. And we can see in orange and blue here that 114 filaments had one polarity orientation and 22 had an opposing polarity. The probability of getting this exact split due to chance is incredibly unlikely, 1.5 times 10 to the minus 16. So given how unlikely that is, it strongly suggests that this is a non-random organization, but must be being driven by an underlying biological mechanism. So that was really interesting. We also investigated tau structure and organization using cryo-FibSem. And we've heard about this before. We can generate lift-out lamella. In this case, we're generating lift-out lamella targeted at regions of amyloid pathology. If we look on the right-hand side at this search map, these boxes are denoting regions that we've collected tilt series. And in orange, we can see regions which contain filaments. In the next couple of slides, I'm going to talk about the tomograms which are inside this yellow box. This one here corresponds to the cryo-clem signal that we can see indicated by the blue arrow. And then deeper into the tissue by approximately 1 to 2 microns, we have another tomogram corresponding to this region here. So these positions were situated approximately 1 to 2 micron apart. They were in separate subcellular compartments because we could identify a delineating membrane around them. When we generated tomograms from these regions, we identified again parallel clusters of unbranched filaments. We wanted to try and confirm the identity of these, so we used subtomogram averaging. In the first of these tissue clusters, we identified paired helical filaments and straight filaments. So this is the first direct evidence that mixed amyloid isoforms can coexist within the same filament cluster in tissue. We also looked at the polarity orientation for paired helical filaments. 33 of these had one polarity, 16 had an opposing. These are in blue and orange. For straight filaments, which are in purple and turquoise, we had a split of 12 and 7. And again, the probability of getting this overall split by chance was low, suggesting that it was non-random. Now, interestingly, when we looked at the other subcellular compartment, situated just a micron away, we only identified straight filaments. And here we had a split of 26 versus 20. So the probability of getting this by chance is actually much more likely. So I've shown you 3 different tissue locations, and it's interesting that in some of those we have highly non-random or highly skewed polarity orientations, whilst in others it appears to be more random or more likely to occur by chance. So we wanted to investigate this further, and one of the factors that we thought could possibly be influencing the polarity orientation is actually the spacing of the filaments within the cluster, or their density. You could imagine if filaments are close enough together, then they may be able to interact with each other. Possibly one filament could be templating the growth of another and influencing the polarity. So to quantify this, we use the particle coordinates along the helical axis of each filament from the central 150 Z slices of the tomogram to ensure that across each data set we're sampling the same volume. And what we found from this was really interesting. In the paired helical filament only cluster, the spacing was significantly less than the mixed amyloid isoform cluster and the straight filament cluster. That's interesting in itself, but also if we look at the correlation with polarity orientation, we found that generally the closer together filaments are, the more non-random these organizations are, and the further apart they become, we're starting to see more likely organizations. So we speculate at this point that this could be indicating that filaments are in some way interacting with each other inside these clusters. So to conclude, we've developed a cryo-clem workflow that enables us to investigate the structure and organization of amyloids inside human brain tissue. We identified that tau forms parallel clusters of unbranched filaments. We were able to resolve the polypeptide backbone conformation to sub-nanometer resolution and identify mixed amyloid isoforms. And through quantifying polarity orientation and filament spacing, we found a correlation which suggests that there could be some level of interaction between filaments that could be controlling how they're oriented. We're currently looking at data from a second donor, and so far the initial data is recapitulating these results. So this has been a huge team effort led by René. Maddie Gilbert is a PhD student in René's lab who's really led the optimization of generating vitreous human brain tissue. And she's also been involved in data processing. Nayab is a postdoc in René's group who's also been heavily involved in sample preparation. And Tom O'Sullivan from the EM facility has been really instrumental in generating those initial Semaviz samples. I also want to thank the Netherlands Brain Bank and the donors who provide their tissue. We wouldn't be able to do this research without that. And also thank you to collaborators in Leeds and further afield. Thank you for listening.
Thank you very much, Joshua, for an excellent talk. There's nothing on Slack, so we'll go straight to the room. I see Stephen has his hand right in the air.
Amazing. What are these filaments doing to the cell? And can you correlate the type of filament to a specific area, mitochondria, like damage? What's going on?
It's a good question. So with A beta, that's outside of the cell. With tau, it's inside the cell. I guess I don't think we specifically looked at correlating it with what kind of other cellular constituents it's around. Looking at A beta, we tend to see what we're calling extracellular cuboidal particles. And we believe that these could be influencing the biogenesis of A beta in tissue.
But do you specifically see damage of membranes or?
Damage of membranes, we do occasionally see that, but it's difficult to determine whether that's caused by how the sample is being prepared or if it's actually a genuine kind of process that's going on. And we don't see it frequently enough that we could kind of have any statistical power over that.
Okay, thanks.
I do have a poster as well, I should say that, 74. So if anyone's got other questions.
Hi. Oh, sorry. Okay. This may be a naive question, so I'm over here. On your left.
On my left, yeah.
Because I'm not in like the neurobiology field, but you said that the brain tissue that you got from the Netherlands had been frozen 2 hours, I guess, after postmortem.
This one was 6 hours.
6 hours,
Yeah,
okay.
6
So
hours.
how do you know that you're not looking at maybe hypoxia-induced changes, for example, in the brain tissue? Can you see maybe you can look for that? I don't know.
Yeah, so good question. One of the things that we've done is use a mouse model, so an NLGF, which has a humanized A beta. And we looked at it fresh, and we also looked at it with a 6-hour postmortem interval. And the morphology and the architecture of A beta is really indistinguishable using methods that we have. Of course, you see changes like microtubules depolymerizing, but we haven't been able to detect any significant changes caused by that interval.
Could the polarity change, or have you looked at that? I don't know if it can.
I think that would be difficult because, as I was saying about when filaments are polymerizing inside a cell, like inside a myelinated axon, if filaments are becoming several microns in length, its ability to rotate 180 degrees is going to be quite difficult.
Great. So that was going to be my question, actually, more on the sample prep. So even though you've got the mouse model and you've treated that in the same way, so you froze it immediately and then froze it 6 hours. Did you do a freeze thaw cycle?
Yeah, we did that as well. Yeah.
So have you also looked, or has anybody looked at with sort of volume imaging? So fixed, resin embedded, and volume imaging of the same tissue?
Not for this tissue, but it's definitely something
Or
that's
similar tissue.
Brain tissue. I'm not entirely sure if anyone's looked at Alzheimer's disease.
Okay, because that will give you far more context of the cell.
Yeah, yeah. I think one of the ideas that we would like to look at is, because we've got repeating sections with some of this, we could, in theory, have high resolution reconstructions from the repeating fluorescence, but it's not something that's trivial to do,
No.
but would be very, very cool and interesting to do.
I'll take one more. We just started with like, yeah, go in the white shadow.
So much this next one. So my questions about the nucleation. Before that, is actually you didn't see any branching of these filaments. That's maybe this explained this in the evolution, because these filaments is pretty young in evolutions, just only exist in Homo sapiens And that's why there's not enough time to evolve to some of the filament binders, especially it looks like the inactive filaments up to 3 like this filament binders to branching. My questions about the focus to nucleation, because nucleation very interesting actually. There are these different nucleated seeds, and so do you think the nucleation does spontaneously happen or any nucleator and already evolve in this Homo sapiens the cells, and how the nucleation, because that's first point, that's most important point.
Yeah, interesting question. I guess the answer to that is I don't know. What we'd like to do is be able to see the start and end of a tau thread or a tau cluster to see what's actually happening at the initiation. But yeah, we don't have that information yet, and we also don't know from this data alone that tau, it could be branched. We don't have that information yet, but everything currently is pointing to the fact that it isn't.
Thank you.
Okay, I think with that we'll let Joshua off the hook and thank him for a great talk.